Welcome to Science with Dr. T. This week, we'll be learning about enzyme inhibition. An enzyme is a molecule, usually a protein, that helps speed up biological reactions. The enzyme has an active site, which is where the reaction occurs, and allosteric sites, which are sites other than the active site on an enzyme. The active site is specific for the enzyme's substrate. So other molecules can't randomly bind because they won't fit in the active site. The substrate binds the active site of the enzyme, then the reaction occurs, and products are generated and released from the enzyme. There are molecules called inhibitors that prevent enzyme reactions from occurring. A competitive inhibitor is able to bind the enzyme active site just like the substrate can, so it can only bind to free enzyme. Remember, the active site is specific for a certain substrate. So in order for the inhibitor to be able to bind the active site, it has to look pretty similar to the regular substrate. Once the inhibitor binds the active site, the substrate can no longer bind. So the reaction does not occur and no products are generated. With non-competitive inhibition, the inhibitor binds to an allosteric site on the enzyme. When the inhibitor binds to the allosteric site, it changes the shape of the active site. The substrate can still bind to the enzyme, which is not depicted in this diagram, but the inhibitor changes the shape of the enzyme so that it is no longer in the optimal position to catalyze the reaction. Non-competitive inhibitors can bind the enzyme alone or the enzyme substrate complex. Uncompetitive inhibition is a little bit different. An uncompetitive inhibitor binds only to the enzyme substrate complex, not the enzyme alone. After the substrate is bound, the inhibitor binds, causing a change in the active site that prevents the reaction from occurring. So the enzyme is inactive. Now, let's put these concepts into practice. Some strains of bacteria become resistant to penicillin through a mutation that allows them to synthesize an enzyme, penicillinase, that breaks the antibiotic down. Which compound is likely a competitive inhibitor of penicillinase? Okay, so we are looking for a competitive inhibitor. This is an inhibitor that will bind to the active site like the substrate, penicillin. So it has to look similar to penicillin since the active site is shaped for the substrate to fit. If we compare the structure of penicillin to our answer choices, the one that looks most similar is B. So that would be our competitive inhibitor. Let's try another problem. Scientists researching the actions of inhibitors A and B on an enzyme noted the results below. Based on the results, what type of inhibitors are these? So inhibitor A has no enzyme inhibitor complex, but it does have an enzyme substrate and enzyme substrate inhibitor complexes. So that tells us that inhibitor A is an uncompetitive inhibitor. Uncompetitive inhibitors can only bind to the enzyme substrate complex, so there would be no enzyme inhibitor complex. Inhibitor B has the enzyme inhibitor complex, which both competitive and non-competitive inhibitors have. It also has the enzyme substrate and the enzyme substrate inhibitor complexes. We know this has to be non-competitive inhibition because a competitive inhibitor does not allow the substrate to bind since they both bind to the active site. So we would not see the enzyme substrate complex. I hope this has been helpful. Make sure to practice on your own with a few different scenarios until you get the hang of it. Thank you for tuning in. Remember to like our videos, subscribe to the channel, and share. Next time, we'll look at Punnett squares.